So we're going to be looking at bone textures. We're going to be looking at compact and spongy bone because we're going to begin our look at the structure of bone by starting with its gross anatomy. Compact bone is dense and it's generally in the outer layer of bone, especially that type of bone that appears smooth and solid. Spongy bone tends to be more of a honeycomb that's internal, made up of small needle-like or flat pieces of bone called trabeculae. It's between these trabeculae and other open spaces that we find either red or yellow bone marrow. We can look a little bit more at these bone textures and note that spongy bone tends to be more internal. You can also see the base structure of trabeculae and how they are pointy structures that can be relatively either rounded or flat. These trabeculae have spaces in between them, and that's where we're going to find things like yellow marrow, which is going to be fat or adipose tissue, or red marrow, which is going to be hematopoietic tissue that allows for the generation of blood cells. Let's compare and contrast just different micrograph views of compact and spongy bone. Here we can see compact bone from the rib on the left and trabecular bone, also known as spongy bone, on the right. In between the trabeculae or spicules of spongy bone, you can see adipose tissue, which is also known as yellow bone marrow. On the left, where we have compact bone, you can see there's a lot less space between the extracellular matrix components of this bone and the osteocytes or cells in the lacunae. So these are just two images of different types of bone viewed with a light microscope. So that gives us an idea of their differences. Let's take a kind of step back and move away from the microscope and really look at bone using macroscopy. So we're going to be looking here at the end of the femur. You can see the bone's quite smooth on the outside, so there's some compact bone that's quite dense. It even gets thicker as you move towards the longer shaft of the bone. And as you move inward, you have a lot of space because that's where you find spongy bone, which is that mesh of spines called trabeculae. And we also see other spaces, like we start to see a cavity down near the shaft of this bone at the bottom of the image. That's going to be the generation or first part of the formation of something called a medullary cavity, where we're more likely to find, especially in long bones, yellow marrow or fat tissue. Now, when it comes to short, irregular, and flat bones, there's a little bit of a difference to their structure. They still consist of compact bone on the outside and spongy bone on the inside, but they tend to have more of a thin plate structure. That thin plate is internal spongy bone, oftentimes called diploe, in short, irregular, and flat bones, and it's covered by compact bone. So it's essentially kind of like a spongy bone sandwich, with the compact bone being the pieces of bread. Both long bones and compact bones have membranes that cover and line both the outside and inside of the bone. The periosteum is a membrane that covers the outside of compact bone, and then you have an endosteum that is an inside membrane that covers the inside portion of compact bone. Throughout the open spaces and surrounding or near these membranes are bone marrow, scattered throughout the spongy bone, and it may or may not be in the defined marrow cavity. You also find the ends of long bones, hyaline cartilage, and it's going to cover the areas of bone that are part of a movable joint to help reduce friction between those structures that move together. Here's a picture of a flat bone. So here you can see the spongy bone is the meat in a compact bone sandwich. You take a closer look at that spongy bone. It's made up of what we can expect of spongy bone, which is the spicules or trabeculae. So still talking about gross anatomy, let's go back to our long bone. Because all long bones have a very typical structure. Not only are they longer than they are wide, they all have a bone shaft called a diaphysis. And then they have expanded ends called epiphyses. In the diaphysis, you have a tubular shaft, that's the long axis of the bone, and it generally consists on the inside of spongy bone surrounding a central medullary cavity or a central space that's often filled with yellow marrow or adipose tissue. Whereas on the expanded ends, you're going to have a much thinner surrounding of compact bone and a lot of internal spongy bone. In most epiphyses, especially those of things like the humerus and the femur in adults, you're going to find red bone marrow. Now, on the ends of these epiphyses, both the distal and proximal end, you're oftentimes also going to find articular cartilage. Articular cartilage is just hyaline cartilage that you find at the ends of long bones. It's leftover skeletal cartilage at flexible areas of the body. Now, keep in mind there's other structures to talk about when we look at the structure of a typical long bone. For example, there's a space between the bone shaft, the diaphysis, and the epiphyses, the expanded end, called the epiphyseal line. Now, an epiphyseal line is a remnant. It's something left over from childhood. In childhood, you have an epiphyseal plate, and that's where bone growth occurs and continues until that epiphyseal plate turns into an epiphyseal line and bone growth stops. So here's our structure for a long bone, and this is actually the humerus of the arm, so the area between your shoulder and your elbow. We can see at the ends, both the proximal and distal end, there is articular cartilage. So there's hyaline cartilage at the end of both sides of this long bone. So we have a proximal epiphyses and a distal epiphyses. The proximal epiphyses is closest to the axial skeleton, and the distal epiphyses is the furthest, but they're both expanded ends. On one side, the proximal epiphyses, we can see we have internal spongy bone. In between the epiphyses and the diaphysis, we can see an epiphyseal line that's completely ossified, completely bone. If this bone was still growing, that epiphyseal line would be called an epiphyseal plate and would consist instead of cartilage cells. If we take a closer look now at the diaphysis, we can also see an area that has an open space called the medullary cavity. It's lined just on its outside by not only an endosteum, but spongy bone. And then we move into compact bone, which is covered on the outside by the periosteum. And the periosteum holds on to the diaphysis using Charpet fibers. These are fibers that penetrate that outer layer of bone. And we also have nutrient foramina that are able to go into bone and help provide nutrition to the many osteocytes of bone. So here is a real bone where we can see the spongy and compact bone orientation. Make sure you know what diaphysis means and where it's located in a long bone. Make sure you know what epiphyses mean and where they are located in a long bone. In terms like epiphyseal line, epiphyseal plate, and articular cartilage, those should not be new to you. You should be very comfortable with the idea of cartilage that is hyaline cartilage at the ends of long bones. The idea that compact bone tends to be more external than the internal spongy bone. That over most bone, if there's not articular cartilage, there's an outer periosteum and an inner endosteum. And that bone, unlike cartilage, is vascularized. It has blood vessels that travel through various different canals and weaves in between the spaces of spongy bone. Now, I've mentioned the membranes a bit, and I just mentioned where the periosteum and endosteum are located. Periosteum on the outside of bone, endosteum on the inside of bone. But let's look at each one more closely. You have the periosteum, which is a double-layered membrane that covers the outside surfaces of bone. It's found everywhere on the outside surfaces of bone except at joints. And if periosteum sounds a little familiar, you might be thinking of the perichondrium, where all long bones were once hyaline cartilage models. And when they changed from cartilage to bone, that dense irregular connective tissue that was on the outside, called the perichondrium for cartilage, it didn't disappear. It just now is outside of bone because the cartilage turned into bone. So we're going to change the name and call it the periosteum because it's no longer surrounding cartilage, it's surrounding bone. So this double-layered structure is just a remnant of the perichondrium. And it has a fibrous outer layer that's just dense irregular connective tissue. And then an inner osteogenic layer that is just going to be right up close to the compact bone. This tends to contain bone stem cells called osteoprogenitor cells, and they can give rise to bone cells, of which we have many different types. We also find that there's many nerve fibers and blood vessels in this osteogenic layer, and they can perforate and move into bone through tiny canals. There's also perforating fibers, sometimes called Charpet's fibers, of the periosteum that hold this inner osteogenic layer to the bone matrix. So it essentially secures the periosteum to underlying bone. And what we can talk about more for the periosteum is what else it can do besides surround a bone. It also can provide an anchoring point for tendons and ligaments. Tendons connect muscle to bone, and ligaments connect bone to bone. And we also have the endosteum. So this is a little bit more of a delicate membrane. So it's a thinner connective tissue, and it covers and lines internal bone surfaces. So it's covering all of that internal trabeculae of spongy bone. It's lining specialized canals that pass through the compact bone, and it is very similar to periosteum. It still contains osteogenic cells that can differentiate or change into other bone cells, but it is a little bit thinner, a little bit more delicate. And what you're probably also noticing is with bone, which used to be cartilage, but with bone, we have blood vessels, we have nerves. Unlike cartilage, which has no nerves and has no blood vessels, bone does have both of those things. Bone is well vascularized, it's really innervated. It has nutrient arteries, veins. These serve different parts of long bones, as our example, including the diaphysis. There's nutrient foramen and foramina that allow for blood vessels to weave in between the structures of a long bone. There's even areas of the epiphyseal spaces that contain arteries and veins as well, and nerves accompany many of these blood vessels into and through the structures of bone. So there was some type of nutrition shift between cartilage turning into bone, because cartilage didn't have these blood vessels and nerves, and now we do have them in bone. When we talk about different types of bone formation called ossification, we'll talk more about this nutrition shift and how blood vessels were able to penetrate this newly forming tissue, which is a connective tissue just like cartilage, but is ossified as opposed to kind of the more gel-like matrix we find in cartilage. So let's do a quick review. We have things like perforating Sharpey's fibers that hold the outer periosteum to the compact bone that is more external than the internal spongy bone. In a long bone, you have a canal in the diaphysis called the medullary cavity that holds usually adipose tissue called yellow bone marrow. We can see in the two expanded ends of a long bone called the epiphyses that you have spongy bone, and those have many trabeculae. And in the case of the example we see on the left, we have a proximal epiphysis separated from the diaphysis by an epiphyseal line. The epiphyseal line is a remnant of the epiphyseal plate. The epiphyseal line is purely bone. The epiphyseal plate would be cartilage, and that would indicate that that bone is still growing. Other things you can see on a long bone are the ends of the long bone that are covered with hyaline cartilage called articular cartilage. So again, that's a lot of information, but it's a quick review of the gross anatomy of bone. Let's finish up with talking about hematopoietic tissue in bone and then bone markings. So when it comes to gross anatomy, we're looking at things both inside and outside of the bone. And red marrow is one thing you could find inside of bone, found within trabecular cavities of spongy bone, or the diploe of flat bones, such as the sternum. In newborns, the medullary cavities, which we're more used to them containing fat or adipose tissue, but in newborns actually contain red marrow. But as we become adults, the red marrow is going to be a little bit more scarce in the body and would only be found in the heads of the femur, in the humerus, and other active areas of hematopoiesis or blood cell generation, like the flat bone diploe of some irregular bones, like the hip bone. And yellow marrow in certain instances, such as when someone becomes anemic, can actually convert into red marrow. So keep in mind what hematopoietic means is blood cell generating. So hemat or hemato is blood, poietic or poiesis is generating. So this is just blood cell generating tissue. And all blood cells, red blood cells, white blood cells, platelets, all of them are made inside of bone in red marrow. All right, and then also kind of finish up looking at gross anatomy, we're going to look at bone markings. These are sites of muscle, ligament, tendon attachment, and they tend to be on the external surfaces of bone. These areas are involved in joint formation, they're openings for blood vessels and nerves. You've probably heard of a couple of them. For example, they might be a certain type of projection, or they might be a surface indentation or a depression or an opening. But these structures are going to be essential for the overall, not only articulation or how the bones fit together, but functioning of the skeleton. So here's some examples of bone markings. A tuberosity tends to be more of a large, rounded projection. You can have small rounded projections that are more of a tubercle. There's trochanters versus tubercles. Some are going to be sharp and slender, others going to be small and rounded. Some of them are really general, like a process is any bony prominence. You also have things like depressions and openings, grooves and fissures, and foramens and meatuses. And the two that I really want you to know the most about are foramen and fossa. Now, a foramen is going to be a round or oval opening, a hole through the bone. A fossa is not a hole through the bone, it's just kind of a depression, an indentation, sometimes called a basin-like depression in bone. It usually serves really as an articular surface or an area where two bones fit together well. So here, for example, we can see many different bone markings, such as the head of the femur, body of the femur, the area where your kneecap sits, the patellar groove. Things like the medial and lateral condyle, which are oriented based on the orientation of the bone in the skeleton at its articular surface. Again, there's a lot of these bone markings, but there are two I want you to know for your lecture exam. Many more will be covered in your lab class. And the two to know are fissure, foramen, and actually let me just add fossa to the three. A fissure is a narrow, slit-like opening. We only want to know that as something different to a foramen. Foramen and fossa are the two main ones to know. Foramen is a round or oval opening through a bone, and a fossa is a shallow basin-like depression. So let's try some practice. All right, here we're looking for something that is a foramen versus something that is a fossa. A good example of a foramen is this big hole on the underside of the skull called the foramen magnum. It's labeled number 7. We also have the indentation or fossa at number 10, and that is going to be where your mandible actually sits into your temporal bone. So here in this view, we're looking at the underside of the skull. So again, that's a quick overview of bone markings. If it seems quick, it was supposed to be, because most bone markings are handled in a lab class where you have more time to look and hold and touch and investigate these bone markings. Next, we're going to be looking at the microscopic anatomy of bone, so make sure to watch that video as well.